Okay, thank you very much. Good morning, everybody. It's an absolute pleasure to be here and to see so many of you. And thanks to Sid, I think he's already gone, for the very warm welcome to Manchester. So my lab's based at King's College London, and I also co-lead the Functional Genomics of Human Brain Development cluster, which is also based out of King's. And what I would like to do right now is to tell you guys a little bit about some of the work my lab has been doing, where we've been trying to think about how we can translate genetic insights into an understanding or neurobiological understanding of complex disorders such as schizophrenia, and how we're taking on some of the lessons we've learned from studies such as this here in the work that we're doing in our cluster. So the main motivation behind our work really comes down to this point that neurodevelopmental and psychiatric disorders prevent a number of children worldwide from reaching typical cognitive, emotional, and motor milestones. And we heard a lot about this here already today, particularly in Sid's talk. And you know, the information that we have received over the last 2 decades in terms of the complex genetic underpinnings of these disorders have really helped us understand what genes may be associated. We've also gone on to understand that in particular neurodevelopmental and psychiatric associated genes are highly expressed during very early stages of neurodevelopment, and particularly in different regions of the brain. And this key stage of neurodevelopment is really important when we consider that it is a really intense period of development where multiple processes are being orchestrated in parallel and that need to be coordinated in order for typical brain development to occur. So the challenge that we see is now, how do we translate the genetic insights into a neurobiological understanding of these different disorders? And to do so in the context of the developing brain, where we have multiple different cell types, different processes occurring throughout the course of development, and that each different brain region and therefore cell type within those regions likely have different molecular logics. So the gene that my group has been studying for some time is this gene called ZNF804A. This gene, or variants within this gene, was the first to be identified as having genome-wide significance for schizophrenia back in 2008. And subsequently, other bigger studies and other genome-wide association studies have identified the same variant as well as multiple others across not just Caucasian ancestries, but also in other ancestries as well, suggesting that this is quite a robust gene that increases the likelihood for schizophrenia. We know from human studies that the variants in this gene are associated with multiple neurocognitive phenotypes. And the original hypothesis was that this gene encodes for a transcription factor, or a gene or a protein that regulates gene transcription, due to the fact that it contains a single zinc finger binding domain. And that's what the majority of studies really initially focused on. But work from our group, as well as multiple other groups, suggested that this may not be the only function that the protein may have, as we started to see that the protein for ZNF804A was not only expressed in the nucleus, but throughout the whole cell, and particularly in neurons at the extremities and along the neurite processes, and even at synapses, the site where communication between neurons occur. So this here raised the possibility to us that ZNF804A is involved in multiple processes. So with this here in mind, a former PhD student in the lab, Laura Sicklinger, decided to ask initially 2 very simple questions. When is ZNF804A expressed, and where is it expressed? Which cell type? And what she saw was that ZNF804A was very much expressed highly during early stages of neurodevelopment. And what we know from studies of the actual variant, that the variant is likely to decrease the expression of the gene, suggesting that it's a loss of function, particularly impacting early stages of neurodevelopment. She further went on to really identify that the main cell type that expresses ZNF804A were these developing upper layer excitatory neurons. It's expressed in other cell types, but this was the main cell type that showed the highest expression for this cell. So taking this information on, Laura then decided to use a human-induced pluripotent stem cell line and to utilize a protocol, a differentiation protocol, that allowed her to generate these developing upper layer excitatory neurons. She used a CRISPR-Cas9 approach to generate not only unedited wild type, but also heterozygous and homozygous mutations, which led to a significant reduction in the expression of the gene at the mRNA level, as well as the protein as well. So she had a very nice loss of function model. So the first experiment that she decided to do was to take these cells and differentiate them into these developing excitatory neurons, and then to basically assess the whole transcriptome. So she carried out a simple RNA sequencing experiment. And what was most striking about this here was that there were actually very few differentially expressed genes. So this was quite contrary to what the early predictions of the function of this gene would be, because it didn't look at that, at least at this stage, at these cell types, ZNF804A was engaging in regulating gene expression. But what we did see was that actually a number of the genes that were being differentially expressed were genes that were associated with adhesion proteins and even synapses. So what Laura then decided to do was to develop and optimize a high content assay to look at synapses by looking at pre and post synaptic proteins, so proteins on either side of the synapse, to look at their abundance and also where they overlap, to use that there as a proxy for looking at putative synapses. And what was really striking here, again, was that the loss of function cell lines, the mutant cell lines, actually showed an increase in the number of putative synapses, which is kind of contrary when we think about schizophrenia, because the majority of evidence suggests that, at least in a end of stage life, in postmortem studies as well as in human PET imaging studies, it is associated with a reduction in the number of synapses. So this here was quite a peculiar, surprising finding, but we went on to then ask the question about whether these cells functionally were different. And we just simply used calcium imaging to look at the excitability of the cell. And we did this here by exposing them to potassium chloride, a depolarizing agent. And what we found was the mutant cells seem to show a greater response to potassium chloride depolarization, suggesting that the cells were more excitable. So they both express more synapses, but also seem to be more excitable. Now, we then were interested in trying to think about, well, what function could ZNF804A be playing in these cells, and how could this be linked to the phenotypes that we were observing? And when you looked at the literature, one thing that really struck out to us was that multiple yeast-2 hybrid studies had identified ZNF804A as being able to interact directly with proteins associated with the regulation of protein translation, as well as directly interacting with ribosomal proteins themselves. Now, the unique aspect about neurons in this context is that a lot of protein translation can occur locally along the distal parts of the cells, along the neurites at synapses, independently of gene transcription. So what Laura decided to do was to develop an assay to try and look at this here, potential protein level changes in an unbiased way. And what she decided to do was to take her cells, differentiate them by growing them on a mesh, which she then enabled her to be able to separate the cell bodies away from the actual neurites. And then she sent those 2 subcompartments off for proteomic analysis. And over here, we can just see how well this approach worked, where you could clearly see a separation of the proteins that are you would expect to see in the cell soma, as well as the proteins that are very much what you would expect to see in the neurites. And when she compared her mutant cells with the wild-type cells, she was able to see quite clearly that there was actually what seems to be a change in the abundance, so not necessarily the actual expression of the protein, but the abundance of particularly ribosomal proteins between the 2 different compartments. And it looked at, on average, there may be more ribosomal proteins in the neurite compartment. And indeed, when she actually complemented the proteomics work with another high-content assay, she was able to see quite clearly that there was more ribosomal proteins along the neurites and the mutant neurons. She further went on to use an assay called Sunset to look at active translating sites, protein translation sites. And she found that, again, consistent with her observations, that there was a higher rate of protein translation occurring along the neurites of these cells. So this really allowed Laura and us to develop this model where we were able to identify the potential role or mechanisms by which ZNF804A was playing in these developing excitatory neurons. Now, one of the things to say here, though, is like, whilst this here really gives us this kind of new insight into what ZNF804A may be doing at that particular time in that specific cell type, it doesn't tell us the whole picture. We know that there are multiple other cell types. So what is ZNF doing in these other cell types? We know that these cell types interact. So the effects that we're seeing, particularly this peculiar fact that there's an increase in synapses, is that they're due to the fact that we're only looking at one cell type. What would happen if we were to bring multiple cells together in more of a system way? And of course, we've looked at one developmental time point and not throughout the overall course. And we know that the effects may change during development. And these cells were grown in 2D. And the brain, I've been told faithfully, is not just in 2D, it is actually in 3D. So taking these sorts of ideas, this allows me to talk a little bit about what we're trying to do in our cluster. And we're still focused on trying to translate the genetic insights into a neurobiological understanding of the neurodevelopmental and psychiatric conditions. But we're trying to address some of the questions that come up from these different studies. And the platform that we're working with is we're taking, again, the approach to identify which genes to look at, so prioritizing them by when they are expressed, where they're expressed, and what conditions they may be associated with, using gene editing to generate particularly heterozygous loss of function cell lines. We're doing this here in iPS cells and human ES cells. And then we're generating multi-region organoids, and we're doing this here at scale and in a reproducible way, including very strict quality control measures, with the idea of being able to bring the regions together in a way that they would potentially do in vivo. And then using a range of different approaches to then interrogate the consequences of these loss of function mutations at a molecular, cellular, and functional level. And just to give you a little taste of what we're trying to do, here we got an example of how consistent the organoids look when you generate them. You can see that they generate the sort of cells and organization that we would hope to see, particularly in organoids. We benchmark them against human fetal tissue to demonstrate that they do look like what they're supposed to look like, and that there's consistency even between multiple cell types and at multiple times. We can fuse them together, and we can even do more exotic studies, such as looking at their function using a multi-electrode array, or even xenographing them into mice. So with that, I would like to say thank you to the people who have done the work. So this work was initially led by Laura Sicklinger with the contributions from many people in the lab. And these are the people associated and involved in our cluster, some of our collaborators. And just to simply say thank you all very much for your attention.